Welcome everyone to Sravika's Biology. In this video, we're going to decode everything about amitosis, what it is, why it matters, and how it's different from other cell divisions. If you are preparing for CSIR NET, pay close attention because this topic is small but often tested in tricky ways. Introduction. Cell division is fundamental for life, growth, repair, reproduction. You're already familiar with mitosis and meiosis, which are highly regulated, precise processes. But amitosis? This is something different. It's primitive, unregulated, and direct nuclear division. No spindle, no chromosome alignment, nothing fancy. It's rare in multicellular organisms and occurs mainly in prokaryotes, senescent cells, or specialized multinucleated cells like placental trophoblasts. Definition. So, what exactly is amitosis? It's a type of nuclear division where the nucleus divides directly, without chromatin condensation, without spindle formation, and without organized chromosomal segregation. The genetic material divides randomly and often unequally. The word amitosis literally means without mitosis. And because it bypasses checkpoints, it's considered uncontrolled. Biological occurrence. Now, where do we find amitosis? It's rare, but important. In prokaryotes, amitotic-like division happens, but technically it's binary fission with FTSZ protein machinery. In protozoa like amoeba, asynchronous nuclear division in stress conditions, leading to polyploid cells. This can help them store genetic material and later complete division when conditions improve. In placental syncytial trophoblasts, they divide amitotically to maintain nuclear number. In cartilage cells, low oxygen, low nutrients make mitosis difficult, so they use amitosis. Also in senescent or degenerating cells as a last attempt before death. Even some tumor cells show amitotic-like nuclear fragmentation when checkpoints fail. Characteristic features. Let's summarize key features. No chromosome condensation, so chromosomes are invisible under a light microscope. No spindle fibers, because no microtubules are involved. Nuclear envelope stays intact or pinches slowly. No checkpoints, CDKs and cyclins are not involved. DNA segregation is random and unequal. Cytokinesis? Sometimes yes, sometimes no, especially absent in multinucleated cells. Energy requirement? Very low because no spindle dynamics. It generally occurs in specialized, senescent, and multinucleate cells. So overall, it's simple, passive, but highly imprecise. Process of amitosis. Amitosis does not have phases like prophase or metaphase, but we can describe it in steps. Nuclear elongation, constriction formation, nuclear fission, cytoplasmic division, which is optional. Step 1. Nuclear elongation. The nucleus stretches into a dumbbell shape. Step 2. Constriction formation. A furrow appears in the center. Step 3. Nuclear fission. The nucleus splits into two. Step 4. Cytoplasmic division. Sometimes happens, sometimes not. End result? Two daughter nuclei, often unequal. Biological significance. Why does amitosis matter? It's considered an evolutionary remnant, a primitive division. It provides a quick response under stress for survival. It's energy efficient, ideal for low-nutrient tissues like cartilage. But it's not used for growth because the genetic distribution is random and risky. Molecular basis. What's missing in amitosis compared to mitosis? No chromosome condensation, so no condensins. No microtubule spindle, no tubulin, no centrosome. No checkpoints, CDK and cyclins are absent. No kinetochore activity, no APC/C regulation. In short, all the high-tech machinery of mitosis is missing. Microscopic observation. Under the light microscope, amitotic nuclei look elongated, dumbbell-shaped, with no visible chromosomes. Under electron microscope, no spindle fibers, no metaphase plate, and chromatin distribution is uneven. Difference from other divisions. Amitosis, direct, no spindle, random segregation. Mitosis, equational, produces identical cells. Meiosis, reductional, leads to variation. Energy cost, low for amitosis, moderate for mitosis, high for meiosis. Amitosis versus binary fission. Don't confuse amitosis with bacterial binary fission. Binary fission is in prokaryotes, highly regulated by proteins like FDSC. DNA replication occurs before fission. Amitosis is random, in some eukaryotes, without a defined mechanism. Amitosis in disease and research. In tumors, amitotic-like nuclear fragmentation may occur due to checkpoint failure, but cancer does not rely on amitosis for growth. In placental disorders, excess amitosis may affect fetal development. Also, amitosis can indicate cellular aging or pre-apoptotic state. Mnemonic. Here's an easy mnemonic to remember amitosis. Amitosis, atypical, mitotic control absent, inequitable DNA distribution, trophoblast cells, occurs in special cases, spindleless, invisible chromosomes. Silent survival method. Key conclusion. Amitosis is not a standard mode of cell division, but rather a fallback mechanism or evolutionarily ancient process retained only in unicellular organisms under constraints. Multinucleated cells with minimal nuclear precision need. Senescent or degenerating eukaryotic cells. Thank you. Your support means a lot. Like, subscribe, and share. Check out our quiz shorts for quick practice questions to test your understanding.